Today I'm going to tell you about methodologies that my lab is employing right now to deconstruct multi-gene effects from chromosomal aneuploidies on phenotypes. So, a little bit of background about me and my scientific history. So, I started off in genetics, doing really classic sort of C. elegans genetics, genetic screens, mutant crossing, all that fun stuff, looking at metabolic gene regulation and redundancy within metabolic networks. And then I moved over to Steve Elledge's lab for my postdoc and got really interested in sort of the chaos that is the cancer genome, which I'm saying cancer genome singular, but cancer genomes are really plural in that every cancer genome is unique. It's a unique combination of mutations, structural variations, and copy number alterations. And so everything I had learned from my classical C. elegans training seemed to kind of go out the window a bit when you look at a cancer genome compared to a normal genome. And so this really got me started thinking about dosage genetics. I feel like this is kind of a better way of thinking about genetics rather than classical Mendelian genetics when it comes to a cancer genome, because obviously genes can occupy many different dosage states and different mutational statuses as well. And this is all combined with many other features happening all at once. And so when I think about chromosome instability as a geneticist, I think about it as really a flexible dial to create complex phenotypes driven by gene dosage alterations. So you may have anywhere, in this triploid background cell line from like 1 to maybe 5 or 6 copies of any given chromosomal region. And this is maybe what the bulk population looks like as it evolves. However, what's happening really in a heterogeneous cancer cell population is that there are many different cells occupying many different copy number states for many different chromosomes. And so natural selection or exogenously driven selection, for instance, under drug selective pressure can act on all these different states present in the population. And the overall bulk population can move more towards deletion or copy number gain, depending on the selective potentials. And so what does copy number alteration, how does copy number alteration create phenotype? Well, it really creates phenotype through changes in transcriptomic output, okay? So here I'm showing you some data from my postdoc paper where we looked at the mRNA log two-fold change for genes on in different chromosomal states across many different isogenic clones made from mammary epithelial cell lines. So these are diploid cells that we induced aneuploidy in, selected a bunch of clones, did a bunch of transcriptomics, and what you can see is that, and this has also been seen by many other people in many other systems, is that generally speaking, the alteration in the transcriptome reflects the alteration in the gene dosage. So imagine a cell with many different genes altered. That's basically what's also happening within the transcriptome. And then obviously, depending on the background of the cell, whether they're diploid or tetraploid, you might get different gene dosage increments based on, whether you get an extra copy in a copy number 4 background, that's a 25% increase, versus an extra copy in a diploid background, that's more of a 50% increase. And so again, getting back to this idea that SYN is really a dial that can dose up or dose down genes on a chromosome. And so thinking about chromosome copy number alterations, and from a genetics perspective, again, it's not quite like a hardwired mutation, but it's also not like quite an epigenetic or transcriptomic effect. It's somewhere in between. It's kind of a squishier kind of genetics. And so during my postdoc, we developed some frameworks to describe chromosomal copy number fitness effects in an unbiased way. And so we did this employing sort of classic genetic framework, but applied instead of to mutations in individual genes, applied to chromosomal copy number. And so we started with diploid epithelial cells that are H-toward or immortalized, but otherwise untransformed. There aren't any oncogenic or tumor suppressor mutations in these cells, as close as we can get to normal cells. We treat them with a spindle assembly checkpoint called reversine, which creates random, mostly whole chromosome copy number alterations within a population. We did this in a mammary and a renal cell background. And then we just let these cells compete in culture and then single cell clone from the proliferatively fit population. And then we use low pass whole genome sequencing to basically count copy number. And so from these studies, what we can do is compare the selected cells after fitness-based competition to the initial population of cells, which were generated mostly randomly. Again, we do have some biases in missegregation, which have been described by others, from Geert Kops to Sarah McClellan's lab, showing that there's some bias towards missegregation of the larger chromosomes. So we do have that bias in the random population. Nevertheless, the final fit population doesn't really look like the initial population. We see sort of a negative selection against a lot of the copy number loss. And then we see potentially some positive selection for certain chromosomal features. And when we compare the selection frequencies in vitro in our assays to missegregation rates, as we also measured in our assays, we see there's really no correlation here. Where we start to see correlation is when we compare our in vitro selection frequencies to the frequencies of chromosomal copy number events in actual human tumor populations. So here I'm showing on the y-axis the selection frequencies in vitro in our two different cell lines and the cognate amplification frequencies of these chromosomes in patient cohorts. And what we can do now that we have lots of isogenic lines derived from these forward genetic screens that are enriched for some of these features that are also enriched in cancer, we can compare their growth rates and we can see indeed, yes, these things do seem to confer some proliferative benefit compared to all other aneuploidies. And also, in some cases, even compared to diploids. So plus 20 is the most common whole chromosome gain in breast cancer. It's also the most common whole chromosome gain, in our mammary epithelial cell screens. If you just let diploids grow in culture, they will eventually gain chromosome 20. It has a slight fitness benefit compared to the diploid. And so more recently, what we've been doing is applying functional genetics in the form of CRISPR screens to aneuploidy phenotypes. And so again, we used our mammary epithelial cell models, looking at mutants that have gain of various different chromosomes, comparing them to diploids in whole genome CRISPR screens. And what we found were some general aneuploidy phenotypes, some of which have been well described. So we get all sorts of proteotoxic or proteasome maintenance genes coming up as synthetic lethal, a lot of RNA maintenance genes. And then we also see our top hits are actually pyrimidine metabolism genes. And so from here, we uncovered that net gain mammary epithelial cells have a sort of inherent nucleotide insufficiency phenotype. So when you lose the ability or decrease the ability to make pyrimidines, that is more detrimental when you are carrying extra chromosomes. Sort of makes sense, more chromosomes, more DNA, more nucleotides are needed, synthesis doesn't quite keep up. And so this might represent sort of inherent vulnerability of aneuploidy, which may already be sort of being exploited through chemotherapies. Okay, so brief summary about what we know about aneuploidy patterns and phenotypes in cancer. So we know that aneuploidy copy number alterations in chromosomes, they're a major driver of transcriptomic variants between tumors and also within heterogeneous tumor populations when you look at the single cell level. We know that aneuploidy creates general stress phenotypes, and there's been a lot of work done on this, including the nucleotide insufficiency phenotype that we discovered, the replication stress phenotype that many people have described. We think that might be tied in with the nucleotide phenotype, as well as the proteotoxic stress phenotype, which is one of the first phenotypes described for aneuploidy. What we also know is that there are most likely CNA-specific phenotypes, and these might underlie, for instance, the tissue-specific patterns that we see in copy number selection frequencies. And so we're curious about how do we identify genes driving these phenotypes, and there's been some work already done in this realm that I want to describe before we get into the multi-gene synergies that I'm going to tell you about. So it turns out that independent gene models like Teresa DeVole, who's giving this lecture series in November, described. So models like Tucson Charm, these are weighted additive dosage balance models, which basically say the following. So they say that the selective potential of an aneuploidy for a given region of a chromosome sort of depends on the sum of the weighted gene effects across that region. And so for a deletion favoring score, this would look like there's more tumor suppressors on a chromosome arm than oncogenes and essential genes. And for an amplification favoring score, this would look like the opposite. So there's more oncogenes compared to tumor suppressors on a chromosome arm. And so these scores were based on loss of function point mutations in the case of tumor suppressors, or gain of function point mutations in the case of oncogenes, or housekeeping annotations and conservation for the essential genes. And so it turns out that this works pretty well. So you can look at pan-cancer frequencies of chromosome, for instance, deletions here. And they are fairly well predicted by these sort of additive gene dosage models, where you just take the tumor suppressors, the oncogenes and essential genes, and you say, which chromosome arms have more tumor suppressors than oncogenes and essential genes. Turns out these are more frequently deleted, and vice versa for genes that have like a low score here, or arms that have a low score. And so generally this works fairly well in a pan-cancer setting where we have lots of information about chromosome amplifications, deletions. We have lots of information about mutations. But this model may not explain selective potentials for certain copy number alterations in other contexts. For instance, it doesn't do a super great job predicting tissue-specific tumor amplification deletion profiles, suggesting there are some chromosome regions that have very tissue-specific phenotypes that don't really overlap well with the mutated oncogenes and tumor suppressors for those cancer types. And then additionally, we're starting to see, as I'm going to tell you a lot about in a minute, that aneuploidies can have very, very specific phenotypes when, for instance, challenged with drugs. And this, again, isn't very well predicted by single gene independent additive models. And so could there be synergistic effects between genes within a chromosome region that may be driving phenotypes that we would miss because individual genes may not be mutated, and we may just not see them altered in any other fashion. And so there are sort of maybe cryptic effects that we're missing. And so something we know from classical genetics and from my time in C. elegans, I'm about to show you an example from my PhD, is that interactions between genes are widespread in nature. So here I'm showing you this example from metabolism, whereby we discovered an alternative parallel propionate breakdown pathway in C. elegans. And so what happens when you knock out genes in the canonical pathway, worms are okay because they have the other pathway. Likewise, if you knock out genes in the other pathway, again, worms are okay. They have the canonical pathway. Now, what happens when you start to mutate or knock out multiple genes across these two pathways? So for instance, when we knock down ECH6, this metabolizes a toxic metabolite called acrylyl-CoA. So when you knock it down, this builds up a little bit. You get a little bit of a sick phenotype, but mostly the worms are okay. They still survive. When you knock out PCCA1 in the ACH6 knockdown background, you see that now they don't have this pathway. They have to go through the alternative pathway. They make a lot of acrylyl-CoA. It can go nowhere. It builds up even more. You get synthetic lethality. Now, what happens if you knock out the upstream gene to ECH6? Well, now you can't make acrylyl-CoA, and these worms actually do even better than when ECH6 is knocked out in the wild type background. And so in this case, this is an epistatic interaction. And so these interactions are really widespread in genetic networks, not just in metabolism, but also in signaling networks. And so we know these things are wired within cells. We know that signaling networks, metabolic networks employ a lot of redundancy, parallel pathways, feedback, feed forward loops. There's all these motifs that are meant to keep the network functioning despite perturbation. And so we wondered whether dosage-driven genetic interactions, such as synergy or epistasis, could potentially be defining some selective potentials for some CNAs. And so what would this look like? So let's say there's this circuit here. It consists of parallel negative feedback loops involving gene A and gene B. So what happens when you lose one gene to a mutation? Well, you lose one of the negative feedback loops, which increases flux through the pathway in one side and the gene B side. But because you've increased flux through the pathway, now you've sort of supercharged the negative feedback loop on the other side, the redundant one. So the end result is basically no change. You can maintain the same output of this pathway. Let's say it dictates proliferation, and the cells are fine. Same if you lose the other gene, because again, you have this alternative feedback loop that kicks in. So you, again, maintain sort of a balanced output from this signaling pathway. But what happens if you lose both genes? So in that case, you've lost both negative feedback circuits. In this case, the pathway can't regulate itself, and you just get constitutive activation of signaling through the pathway. So what would this look like mathematically? So we can take a page from drug interaction playbooks, which is to look at bliss independence scores. So basically, this is a multiplicative model. So we would expect potentially that the effects of loss of gene A or dosage reduction of gene A, dosage reduction of gene B might be a multiple of each other. However, if the observed effect of dosage reduction of both of these genes is greater than the effect we predict from multiplication of the effects, then we can call this synergy. Okay, and in this example, that's exactly what we would see, because essentially the phenotype from loss of one gene or the other is essentially zero, 0 times 0 is 0, but we get some massive effect from loss of both genes. Okay, so what would this potentially look like in actual data? So this would look like CNA-driven phenotypes that are not well predicted by single gene effects or gene additive models. So for instance, let's say you have a phenotype, again, cancer drug resistance. I'm foreshadowing this a lot because I'm going to talk about it soon, I promise. So let's say that you have a lot of in vitro CRISPR screens looking at drug resistance for a specific cancer cell line. For that cancer type, we also have patient mutational data longitudinally, so we know which mutations are associated with acquired resistance in the clinic. Let's say that some of those genes overlap. So these would be the single gene effect drivers that can affect the cancer response to that drug. Now, let's say you also observe that an aneuploidy can give you the same phenotype. However, the genes on those chromosomal aneuploidies that give you the phenotype are not overlapping with these genes that you find in the CRISPR screens or mutated in patients. This might be a perfect candidate for a synergistic gene effect dictating this phenotype driven by aneuploidy. And so this has been observed fairly recently in yeast, interestingly also in the realm of drug resistance. This is work from Yuan lab, and there's a lot of work from Chris Campbell's lab showing that aneuploidies in yeast that have specific phenotypes can often be driven by genetic interactions, either in this case between genes on the same region or between genes on different chromosomes that are co-amplified or co-deleted. And so in yeast, very powerful genetics. There's a lot of work being done in this realm there. So we wondered whether this could also be true in humans. And so what do we know about chromosome instability and drug resistance in human cell lines and human cancers? Well, we know that it is very much involved. So there's some great papers published in Developmental Cell in 2021 from Jason and Stefano's lab, showing that if you increase chromosome instability by, for instance, messing with spindle assembly checkpoint, creating more aneuploidy in the population, this accelerates the evolution of drug resistance, both in cancer and non-cancer human cell lines. And what these groups also noticed in these papers is that oftentimes what you end up seeing in the drug-resistant population are new or different enriched aneuploidies, suggesting specific aneuploidy drug interactions. Okay. So my student Faith has been wondering whether CNA fitness landscapes change under drug challenge and whether this results in distinctive patterns of copy number selection that we can observe over big data sets or over meta-analysis of many individual drug resistance experiments that have been done across labs. And so she's compiled a whole lot of data. She started with the PRISM data set. The PRISM data set is a large-scale drug cancer cell line interaction data set. It's basically a pool of cancer cell lines that are barcoded, for which we know the genomes of the cancer cell lines and all the mutations as well in those cancer cell lines. These populations are treated with different drugs. So it's hundreds of cell lines versus, I think, hundreds of drugs, maybe thousands at this point. And so basically for every cell line, you can derive a profile for every drug in terms of its response to that drug or its resistance to that drug. And so, for instance, when we're looking for copy number alteration interactions with drugs in this data set, we've been basically looking for correlations between specific chromosomal copy number alterations and drug effects. So for instance, in this mock data, you might see a correlation between the copy number alteration of chromosome A and dabrafenib, which is a BRAF inhibitor. Now, if we start looking at other RAF inhibitors in the data, we might see something like this, which is to say the same copy number alteration correlating in the same way across all the cancer cell lines. And so what we did was we grouped drugs into drug sets and then performed essentially a gene set enrichment analysis only using drugs to identify drug sets that were strongly associated with certain chromosomal copy number alterations. So this is what this might look like for those RAF inhibitors. You have each of them correlating strongly with chromosome A copy. And so if you rank all the drug effects for their correlations to chromosome A, you see that the RAF inhibitors are all piled up on one side of that ranking. And so for every chromosomal arm, we perform this analysis across all drugs and drug sets. And this is what that analysis looks like. So you can see already there's like a lot of structure in this data. Anything with a color here indicates a significant correlation by FDR. We have all the chromosome arms along the x-axis and along the y-axis. We have all the drug classes for which at least one arm scored. So you can see already there's a lot of interesting things happening here. I'll bring your attention just to these squares here, which are suggesting that loss of chromosome 13q might be associated with MAP kinase and RAF inhibitor resistance. And so we started looking at any data set we could find from the literature whereby someone threw a RAF inhibitor on a cancer cell line and then did some sequencing, RNA, DNA, whatever. We took whatever we could get, and we started seeing this pattern emerge over and over and over again, whereby resistant cells tended to acquire loss of chromosome 13 relative to the naive cells. So this is in C and V plots from some single cell data, looking at evolving populations under dabrafenib, which is one of the BRAF inhibitors used clinically. And here we see some DNA sequencing showing that this population of naive cells already had some pre-existent -13 clones, but these took over in the dabrafenib resistant population. So we also found data sets that did in vivo experiments. We see the same thing. We see that in xenograft models of BRAF inhibitor resistance in melanoma, that tumors that emerge with acquired resistance also tend to lose chromosome 13, among other things. And so we summarized all of the individual experimental data sets that we could find and compared those to our PRISM correlations. And we see what really sticks out is loss of chromosome 13. And also, I should say, one of these studies was from the Jason and Stefano papers, in which one of the drugs they used was a BRAF inhibitor in a melanoma cell line, and they do in fact see loss of 13. So this is very, very robust. But of course, we wanted to validate in our own hands that we could see this. And so we did our own forward genetic copy number screen under vemurafenib selection in A375 BRAF mutant melanoma cells. And what we find indeed is an enrichment for loss of chromosome 13. I'm just showing you one rep of one experiment. We've done this many, many times. We've done different doses. We always see loss of chromosome 13, and we see some additional loss of chromosome 3. So let me back up and tell you a little bit more about BRAF, about the RAS-RAF-MEK-ERK pathway, and about resistance to this drug. So BRAF inhibitors have been used clinically in BRAF mutant melanoma for over a decade now. They have really great initial responses in patients, but then almost every patient will invariably develop resistance to the drug eventually. So BRAF inhibitors are targeted inhibitors of the mutant BRAF. Currently in the clinic now, they're usually combined with a MEK inhibitor because MEK mutations, gain-of-function mutations, are often found in acquired resistance in patients. So this sort of cuts off one avenue of resistance. Invariably, many patients also relapse on the dual drug treatment. And from several studies looking at genomics of patients pre and post treatment failure, we see that gain-of-function mutations in these orange genes are found, or loss-of-function mutations in the blue genes, in about 60% of patients, sort of explaining the resistance. But the resistance mechanism is unexplained, meaning none of these genes are mutated, in about 40% of cases. And so we're wondering whether copy number alterations could potentially contribute. Unfortunately, we don't have good data on that from the clinic yet. And so we started looking at CRISPR screens for vemurafenib or BRAF inhibitor resistance, of which there are many CRISPR screens. So this is actually, I think, this is a Feng Zhang paper. It was like the first CRISPR screen ever. It was actually for vemurafenib resistance. And so what we see over and over again from all these screens is sort of the same core set of hits that, when knocked out, confer vemurafenib resistance. Okay, great. And some of these genes, including the NFs, are also mutated prominently in acquired resistance in actual patients. So none of these genes are on chromosome 3 or 13. Also, no chromosome 3 or 13 genes are recurrently mutated in acquired resistance in patients. And so we hypothesized that maybe chromosome 3 or 13 may contain genes that are acting synergistically to confer BRAF inhibitor resistance. And so we checked the downstream output of the RAS-RAF-MEK-ERK pathway, which is phospho-ERK, in cells that are drug naive but have lost chromosome 13 or chromosome 3 and chromosome 13. And what we see is that, let's just look at wild type cells. These are just two replicates of the experiment. We see, like we should, phospho-ERK depleting with BRAF inhibitor treatment. We see that's also true in the -13 and the -3 and 13 cells. However, to a much less degree. So we still see some phospho-ERK in the vemurafenib condition, whereas we really don't see much at all in the wild types. And there's also an increased baseline phospho-ERK in these cells, and this is all quantified here. So in some ways, it looks like, the -13 cells under BRAF inhibitor resistance, they're almost phosphorylating as much ERK as the wild types under normal conditions. And so this results in a higher proliferation rate under vemurafenib, which I'm showing here for both chromosomal losses. So we found that loss of chromosome 13 cells respond fundamentally differently to BRAF inhibitor at the transcriptomic level. So here I'm showing you on the left some gene set enrichment analysis of comparing -13 clones to the wild type parental under baseline or plus vemurafenib conditions. And so under baseline conditions, there's not really a huge amount of difference between the -13 cells and the wild type cells. You really see a large difference only when you add the drug. And we see down regulation relative of some stress pathways and upregulation relative of the sort of pro-growth signaling pathways. And so here I'm showing you some raw data showing that these are some cell cycle regulated genes. So in the naive clones or the wild type clones, we see that upon vemurafenib treatment, you lose the expression strongly, almost down to nothing of these cell cycle genes, meaning the cells have stopped cycling. However, cells that have lost chromosome 13, each dot here or each column here is a different clone. So we've done this in lots of different clones. We see that these genes do not decrease as much. So there's still residual cell cycle happening in cells that have lost -13. So how are we going to identify the genes that are contributing if we think that they are synergistic? So we settled on a targeted combinatorial dual guide CRISPR screening approach. And so we had to pick genes on the chromosome. We couldn't do the whole chromosomes because there's too many genes. The combinatorics get crazy. We would be screening until the end of time. So we had to narrow down our list of genes that we were going to study. And so we narrowed this down based on genes that were located within regions of chromosome 13 that seemed to be deleted most frequently in our screens. Turns out this was a bit of a red herring. That's this locus here. We saturated this locus. Turns out none of these genes are hits. This is just a very fragile region. We also included genes located broadly on chromosome 13 that had been implicated in drug resistance or pro-survival signaling in the past. Any gene that scored even weakly in at least one published genome-wide CRISPR screen for BRAF inhibitor resistance was included. And we also expanded our search using gene co-essentiality networks. So for every gene on the chromosome, we looked in DepMap. We found the genes that were most strongly correlated with that gene. And then we did gene set enrichment, looking to see if that gene set was found to be enriched in the CRISPR screens. And we also included genes that had paralogs on the chromosome in case it was a paralog type of situation. And so we cloned this dual guide CRISPR library in the following manner. We used Gibson assembly. We used essentially the same guide pool for both positions, the human U6 and the mouse U6 promoter, as well as different scaffolds were used to sort of try to prevent recombination. So first we cloned in the guide pool 1 into this purple locus here. And then we took this pool and we cloned in essentially the same pool of guides into the second position. And so we have an all-by-all combinatoric library. We included cutting controls and we included 3 guides per gene, as well as some positive controls that have come up in other CRISPR screens that we know are important in BRAF inhibitor resistance, which are NF1 and 2. And that led to a total of about 22,000 different combinations. So we infected A375 Cas9 expressing cells with this dual guide CRISPR library. After puromycin selection, we split these cells up into DMSO only or vemurafenib treated arms. And then we compared the populations at each time point. And so just as a quick idiot check, we also threw this library into cells that were not expressing Cas9. We see that under vemurafenib that these cells don't grow at all. When you have Cas9 in the cells, we do see resistance. So this suggests that we are hitting some genes in our combinatoric library, including hopefully the positive controls that are conferring resistance. And then this is just without vemurafenib. Obviously, they grow much faster. So again, checking our positive controls, these enrich both very robustly under vemurafenib. One thing we did was we sequenced the genomes of the population of cells initially and at the final drug selected population. And what we see in both replicates of the screen is that we are getting some loss of chromosome 13, which we might expect given the fact that we know loss of chromosome 13 is also going to confer resistance. So we might be like sort of fighting against sort of natural resistance that's accumulating in the population. We're also just shredding chromosome 13. So we might be helping that along a little bit. So we estimate that about 15 to 20% of the population has lost chromosome 13. That doesn't invalidate our screen or our analysis, which I'm going to show you next, because it does create some potential noise, but still about 80% of the population should be resistant because of the guides that we've put in and not because of the chromosome lost. Okay, so here's what the data looks like. So here I'm showing you each square is a pairwise combination of all the different genes in our library, summarized from the 3 guides for each gene. And so first, let's look at the negative control. So these are the pairs with a cutting control that targets the AVS locus or other loci in the genome that don't encode genes. And so we see very little red or blue, meaning enrichment or depletion, in these essentially single gene knockouts or knockdowns. However, we do start to see in this cluster down here, enrichment or conference of drug resistance, but only when these genes are combined together. And there are really 3 interactions that strongly stand out. One is this a model PDC-D10 combination, a model LATS2 combination, and SPRY2 and LATS2. And then there are other genes that seem to sort of be boosters for these genes, but don't score as strongly. And so let's look at a protein-protein interaction network. This is generated from BioGrid data showing all the genes that we tested in our library and their connection to genes that are important in RAS-RAF-MEK-ERK pathway and genes that are important in resistance to RAF inhibitors. So the blue and orange genes are core genes in the pathway that have been found to be mutated in resistance. We have our positive controls from the screen, NF1 and NF2 here. They both score on their own, as expected. And then all the other genes in pink and blue are the genes tested in our screen, and they're colored by chromosome location. So let me just highlight those 3 prominent pairs that we found. So one is the SPRY2 LATS2 combination. Both of those are on chromosome 13. We have in a model LATS2 combination that's across the 2 chromosomes. And then in a model PDC-D10 combination, those are both on chromosome 3. And here's all the other genes that scored weakly in combination with those stronger hits. And so you can see these are the genes that are most connected to the pathway, sort of reassuringly. And so now we wanted to learn a bit more about ERK signaling as it actually happens in the cell. And this is a very complicated pathway described mathematically by Peter Sorger's lab in 2020 in a very nice manner. So the RAS-RAF-MEK-ERK pathway actually contains a lot of negative feedback, and that results in this sort of pulsatile behavior of phospho-ERK activity in cells. And so this model was very well and carefully parameterized by the Sorger lab. So here I'm showing some predictions on the top from the modeling for ERK signaling under various BRAF inhibitor conditions and MEK inhibitor conditions. And you can see in the actual experimental data, this matches up fairly well. So this model is very good and very powerful. Mathematically, kind of disgusting. I won't show you, but needless to say, they've done a really good job annotating it. And so it's very easy to use. And so what we wanted to do was test whether if we dialed down some of the genes that are participating in this pathway in a negative sort of feedback motif, such as SPRY2, and LATS2, which we actually had to include in the model as a regulator of NF1 and 2. We wondered whether if we dialed them down, would we affect phospho-ERK signaling output? And so what I'm going to show you is a simulation here where we're starting off with no EGF. We're going to drop in a pulse of EGF, and we're going to watch the dynamics of these various genotypes in the model. So here, without exogenous EGF, the cells are entirely relying on the mutant BRAF to signal independently of the ligand. And so at low doses of BRAF inhibitor, the cells are signaling fine. At the higher doses, they're completely shut off. All right, so let's add the EGF and watch what happens. So here's our wild type cells, and here's our double mutant cells, and here's our single mutants. So what we see is that after the ERK pulse, the EGF pulse, sorry, we see different dynamics in the phospho-ERK to total ERK output, depending on these different genotypes, whereby the single mutants have a relatively small effect. The double mutant has a much larger effect, and this would be synergistic. And one thing I wanted to point out here, if I can just manipulate this a little bit, is that actually the double mutant inherits two different dynamical properties from the single mutants. So at early time points, the double mutant looks more like the SPRY2 mutant, so it has a much higher pulse of activity initially before the negative feedback kicks in. Once the negative feedback starts to kick in, we start to see that the double mutant is behaving more like the LATS2 mutant, and that it has a slower dynamic return to baseline. And so from inheriting both of these features, it ends up with much higher phospho-ERK signaling after it reaches steady state. Okay, so now we want to test whether these genes might really be driving this phenotype and synergy. And so we've started to generate single and double mutants using CRISPR. We won't want complete nulls. We want this to mirror a dosage reduction. And I think we've more or less achieved that. So looking at the LATS2 protein levels in our LATS2 knockdown and our double knockdown, we see that they're decreased. And it's the same when we look at the SPRY2 protein and the SPRY2 single and SPRY2 LATS2 double, we see decreased SPRY protein. So we also checked whether we wanted to verify that we weren't losing chromosome 13 in these mutant populations, and we find that chromosome 13 is stable. So now we have loss of one and both of these genes without chromosome 13 loss, and we can really test the individual and synergistic effects very carefully. And so these validations are still in progress. Sorry, I don't have the nail in the coffin experiment here, but I will say that the double mutant is looking very promising as phenocopying the -13 phenotype. And so some of the things that we're looking at are ERK phosphorylation in the single and the doubles, growth rates and various doses of EGF and BRAF inhibitor, and also we're testing some of the predictions that the pathway about the pathway dynamics from the modeling under various EGF and BRAF inhibitor doses. So one thing that the model also predicted was that the double mutant would also be resistant to MEK inhibitors. So here I'm going to show you a same kind of experiment that I showed you with the curves, but now this is a drug matrix experiment where we have the BRAF inhibitor along the y-axis increasing and the MEK inhibitor along the x-axis increasing in the wild type, the singles, and the double mutants. And what you can see, we'll post an EGF. So the brighter colors equals more phospho-ERK. Again, the double mutant is inheriting different dynamics from the singles and ultimately showing higher phospho-ERK levels at all different combinations of RAF and MEK inhibitors. And this is really important clinically because right now in the clinic, people get both of these drugs. And we have some sporadic evidence that we've found from single-cell data in the literature, inferred from single-cell RNA-seq for loss of chromosome 13 in acquired MEK inhibitor resistance. This is an in vitro study. We see, again, in the resistant cells and the trametinib resistant cells, a loss of chromosome 13. And we see potentially some loss of chromosome 13 also occurring in acquired MEK inhibitor resistance in vivo experiments. So we will also be testing MEK inhibitors. So just to summarize what we think is going on with loss of chromosome 13, we think that this is driven by a synergistic multi-gene effect, and this is creating the selective advantage. And it's via a loss of redundant ERK pathway negative feedback loops governed by LATs and SPRI. So under normal circumstances, this pathway can pulse because it's got, after initial EGF signaling, it's got two redundant negative feedback loops. And then when you lose one or the other side of the feedback loop can compensate, and you end up with sort of the same baseline ERK activation proliferation. However, when you lose both genes or dial them down dosage-wise, you lose both feedback loops. The pathway cannot negatively regulate itself, and you get rampant ERK activation and proliferation, even under BRAF inhibitor. But one thing the model predicts is that this will be EGF dependent, so we are also testing that. So one other conclusion from my talk is that chromosome-specific dual guide CRISPR screens can actually reveal these kind of genetic interactions. So I showed you this slide before. A synergistic effect from a CNA might look like no overlap in genes that are known to have single gene effects. However, we may be able to identify some of these using these kind of dual guide libraries. And with that, I will just thank my lab, especially Faith, my student, who's done most of this work. She's gotten some help from our RA Abdul, and also from a summer student and a couple of rotation students. And I'll also thank Decker's lab and Mike Lee's lab in my department for helping provide reagents, cell lines, a lot of things that we've used for these studies, as well as Yolanda Van Leeuwen, who's a real genetic interactions person. She studies genetic expressors, and she joined our department a couple years ago. She's been helping me think about, how do we quantify synergistic effects? What if there's more than two genes? Do we add synergistic effects? There's all sorts of questions that we don't know how to address yet mathematically. She's sort of helping me think about that. And also big thanks to Jason Stefano for really kicking off the field of chromosome instability and drug resistance, and providing a lot of good feedback for our studies and my funding. And with that, thank you for listening.
Thank you very much, Emma. That was a terrific talk. So everyone in the audience, please write your questions in the chat, or just basically raise your hand if you want to ask Emma directly. So we'll get. Thanks, Sabina, for writing that as well. Any questions from the audience for Emma? Stefano, yeah, please go ahead. Thank you.
Hello, hi everybody. Emma, that was beautiful. I was thinking if you have tried or if you thought to use inhibitor of KRAS for the second part of your work.
We have not messed with KRAS yet. I think that would be really interesting though, because so far we've really been focused on BRAF. We have some inclinations that the MEK inhibitors will also be interesting to look at. RAS, we don't know. So that is something that I think would be really cool and we should probably do. But yeah.
Happy 4th of July. Thank you.
Oh, thank you.
So Emma, I have one question. So it's just kind of more theoretical, perhaps, but I mean, obviously, quantifying interactions is an incredible way forward to understand drug resistance mechanisms, as you have illustrated very well. Do we have enough data to basically explore that at the scale? How powered are we? I mean,
not at all. Not at all powered. So this is why we've picked a phenotype associated with a copy number alteration for which there is a tremendous amount of data. first of all, validating that this copy number alteration does confer drug resistance. But also, there's a lot of individual gene data in the form of CRISPR screens. I think there's been like 5 or 6 CRISPR screens performed for vemurafenib resistance and BRAF mutant cell lines. And we have a lot, relatively speaking, of patient data. Now, that's to say, we have good mutational patient data. We don't have good copy number data from patients. So I think that as more data comes online, we will be able to do more of these kind of very targeted studies trying to identify synergistic effects. We also are, we're very limited with how many genes that we can utilize in these kind of screens. Like I said, we ballooned from like 50 genes into like 22,000 reagents. So, it scales exponentially. So you have to be very careful about which genes we pick. But the good thing is once we find some hits that have some interactions, we could design, for instance, like LATS2 and SPR2, we could take LATS2 and now target all the other genes on chromosome 13. That would be very feasible. So we can sort of like, start sort of sparsely looking at the interaction space and then maybe expand on certain regions of that interaction space.
Right, nice. I mean, I was also thinking that we tend to consider CNAs as chromosome gains and losses, right? But there are obviously other events that are becoming more, we are realizing they're more frequent, such as chromotrypsis and the like, which would make the basically cell look like a diploid, for example, right, in single cell RNA sequencing data, but not necessarily all the genes will be wild type in there. So I don't know if you have any thoughts about those effects as well.
Yeah, I think we probably again need a lot more data, and we need better ways of integrating potential effects from copy number and also the potential effects from mutations and SVs, which are going to generate a lot of new mutations or gene expression alterations. So yeah, it's a bit of weeding through all that chaos and trying to best capture all the effects that might be happening within a cell population, quantifying those effects and determining whether they're additive or synergistic. Yeah, it's a big challenge for sure. I think this is why we've spent a lot of time on one very specific example.
No, a very important one indeed. Yeah. Great,
Just to
thanks.
show what's possible. Because I think if we can just show, these things are happening, now we have to start looking for them, right? So
No, indeed. Thank you. Any more questions for Emma? Orly, yeah, please go ahead. Thank you.
Beautiful talk, Emma. I was just wondering, are you looking into sex-specific differences in response to treatment for cancer patients?
that we have not touched yet. Yes, but that is a very interesting possible variant factor, for sure. No question. So again, this comes down to like, power, whether we can do all the analysis, splitting up the groups into male and female, whether we have matched numbers of males and females. Obviously, breast cancer is very female dominated, prostate's very male dominated. So yeah, like I think in a perfect world, we would have male and female versions of our cell lines, but we don't. But that's a good point.
Thanks.
Thank you. Any more questions? If not, we will let Emma and all our US colleagues enjoy the 4th of July weekend. And yeah, I wish you, all of you, like a nice summer, and we'll see you in September at the lecture series. Thanks everyone for joining, and thanks so much, Emma, for a terrific talk. Bye-bye.
Thank you.